Swayam Prabha. Digital India. Educated India.
Hello everyone. Welcome to the next lecture of the second unit of Plant Biotechnology course. In this unit, we are talking about the production of haploid plants from male gametophytes or the female gametophyte, chromosomal doubling, chromosomal elimination techniques, and so on. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering at the School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. This project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. In this lecture, I will discuss about the production of haploid plants through in vitro gynogenesis, that is, the female gametophyte, various factors that affect the gynogenesis, then what are the major challenges for the improvement of the technique of gynogenesis or its establishment in the plants where it has not been done yet, or the plants which are recalcitrant for the gynogenesis, and finally the various applications of the gynogenic haploids. Gynogenesis, how we define it. Earlier we talked about androgenesis, where haploid tissue from the male gametophyte, that is pollen, were used as explants for the in vitro cultures and haploid plants were regenerated. Similarly, the gynogenesis is associated with the culture of haploid tissue from the female gametophyte. The process of development of haploid plants by in vitro culture of unfertilized egg cell or any other haploid cell from the female gametophyte is called as in vitro gynogenesis. So here is the general structure of a typical flower. A flower has mainly four parts, that is calyx, corolla, androecium, and gynoecium. The outermost whorl is made up of sepals and collectively called as a calyx. Next to sepals are the petals, which are usually colored, and collectively these are called as corolla. The third whorl is androecium, that is the male reproductive organs. It is the collection of stamens that have two parts, filament and anthers. Earlier in the first lecture, we talked about the use of anthers or the pollen grains that developed or formed within these anthers for the in vitro culturing to raise the haploid plants. And the term associated with the pollen culture is the androgenesis. Finally, in the center of the flower is the gynoecium. That is the female reproductive organ carpel. Usually a carpel have three well-defined parts. The swollen vase is called the ovary. It contains ovules where the female gametophyte develop. A stalk-like structure above the ovary is called as style, and the uppermost part, which is sticky and can have different shapes in different flowers, is called as stigma. The pollen usually land on the stigma and germinate here. Mature female gametophyte in angiosperm flower is usually a 7-cell structure which are organized in a specific arrangement. Let's have a look on the structure of ovule and the female gametophyte. So here is a typical cartoon structure of the ovule. Usually it is an oval-shaped multicellular structure which is present inside the chamber of the ovary. One ovary can have either one or more ovules. A mature ovule consists of two parts, usually the funiculus and the body. Funiculus is the structure that connects the body of the ovule with the ovary wall. And this upper part is called as the ovule body or the body of the ovule. Ovule body is made up of three major components. That is the integuments that forms the outermost layer, which is protective in nature. Next to the integument, second part is the nucellus. It is the remnant of the megasporangium in which the female gametophyte development starts. And finally, the embryo sac, that is the female gametophyte. Ovule body has two defined ends, that is the micropylar end and chalazial end. A small opening at the tip of the integument is called a micropyle, and this end is called as a micropylar end, and the opposite end is called as the chalazial end. Usually the pollen tube with the male gametophytes penetrate through the micropyle, and later the male nuclei fuses with the egg cell, forming the zygote. A female gametophyte in angiosperms is made up of 7 cells that had specific arrangements and specific developmental roles. The 3 cells towards the chalazial end of the embryo sac are called as the antipodial cells. In the center, the 2 nuclei fuse to form the 2N nucleus, which is also called as secondary nucleus, or at the maturity it is also called as the central cell. And finally, the 3 cells that are present on the micropylar end, the central cell among these 3 is called as the egg cell, and the cells on the side of the egg are called as the synergids. Under normal circumstances, the pollen land on the stigma and germinates. The pollen tube grows through the style and finally reaches the female gametophyte through the micropylar end. It enters the embryo sac through the one of the synergids and release the 2 male nuclei inside the embryo sac. One of these nuclei fuses with the egg cell and form the zygote that ultimately develop to form an embryo, whereas the second nuclei fuses with the central cell nucleus which is already 2n, and the fusion of 1n male nuclei make it 3n or triploid that develop into an endosperm. Now, in some plants, egg cell can divide independent of any stimulus from the male gametes, that is without fertilization. And this spontaneous production of haploids from the female gametophyte have been reported in nature, and the process is called as the apomixis. However, the fertilization independent development of egg cell can be induced through the application of the external stimuli too, and is called as the in vitro gynogenesis. Low temperature shock is one of the most successfully used external stimuli for this purpose, but other treatments have also been used, and we will talk about these treatments in more detail little later in this lecture. Development of in vitro regenerated gynogenic haploid plants was reported in 1976 for the first time by Saint Noem and colleagues in Worley. And since then, gynogenic haploids have been developed for several other commercially important plant species like onion, sugar beet, brassica, melon, sunflower, mulberry, rice, potato, and so on. There are several factors that directly affect the successful establishment of gynogenesis or production of gynogenic haploids. Broadly, these can be categorized in the 4 categories. These are the genotype of the donor plants, explants or the developmental stage of the gametophyte, pretreatment required for the induction of gynogenesis, or the composition of the culture medium. So the first is the genotype of the donor plants. Earlier we talked about how the genotype of the donor plants can impact their in vitro regeneration potential or the androgenic potential. Similarly, the genetic makeup or genome of the donor plants has a significant impact on achieving the successful in vitro gynogenesis. Some plants may easily respond to the in vitro cultures, whereas others may be recalcitrant and may not respond to the existing protocols. Interspecific variations in the gynogenic potential have been observed in the various species of genus Allium, that is the onion. It is not only the genotype of the different plant species or different group of plants that have the genotype specific impact on gynogenesis, but the 2 cultivars or the accessions of the same species also can have different responses towards the gynogenesis. For example, the variations in the gynogenic potential of various genotype or accessions of onion, sugar beet, and maize has been observed and reported in the literature. Next is the explant. Now, explant is the tissue or the organ that are used to initiate the in vitro cultures. And so the physiological status or the health of the explant is also very important, that can impact the successful gynogenesis. Now, depending upon the size of the flower and the ease of extraction of female gametophyte, either ovules, ovaries, or the flower buds can be used as explants for in vitro gynogenesis. Flower buds and the ovaries have lots of sporophytic tissue, and callousing from the sporophytic tissue may result in complications. Though the ovule too have the sporophytic tissue, however, as it is very difficult to excise the female gametophyte for the in vitro culture, therefore ovules are the ideal explant in this case. Here is the typical cartoon structure of the flower, and the female gametophyte is present in the ovary. So typically, if we are using a flower bud as an explant to initiate the gynogenesis, it would contain all the flower organs like sepals, petals, androecium, and the complete gynoecium, which are part of the sporophytic generation, and their ploidy level is the ploidy of the parent plant. If it is possible to extract the ovary from the flower, that is this swollen base of the gynoecium, most of the sporophytic tissue could be removed, like the petals or the stamens or the sepals or the part of the gynoecium also. Further, if it is possible to remove the ovules from the ovary, it could help to significantly reduce the sporophytic tissue related complications in the in vitro culture for haploids. However, it is tedious and difficult to extract the ovules, and usually may not be possible in many of the plant species where flowers are really small. The female gametophyte develop inside the ovule. Ideally, the female gametophyte would be the best explant source for the gynogenesis. However, in the absence of easily accessible tools for isolation of the female gametophyte from the ovule, it is not feasible, and therefore ovules are the ideal explant in the current situation. Maybe in the future, with the technological developments, we would be able to extract the gametophytic cells from the ovules, and we would be able to use them as an explant to initiate the in vitro gynogenesis. Here is a structure of mature embryo sac, which is a 7-cell structure, and all the cells are organized in a specific arrangement, like the antipodal cells on the chalagial end, then the central cell, which is usually in 2N or the diploid, and then 3 cells on the micropylar end, one of which, or the central one, is called as the egg cell, and the cells on the site of the egg are called as the synergids. The development stage of the gametophyte is very important for the induction of gynogenesis, and usually the mature embryo sacs are more suitable for this purpose. For many plants, it has been observed that even if the ovules are harvested at earlier stages of the female gametophyte development, its development continues even after its excision from the parent plant while it is cultured in the in vitro culture, and it continues until all its constituent cells have been differentiated, that is, mature embryo sac stage has reached, and then it can be induced to develop the gynogenic plants. Let's have a quick look on the development of the female gametophyte in angiosperms. The female gametophyte develop inside the ovules. So each ovule typically have a single megaspore mother cell. Megaspore mother cell undergo meiosis to generate 4 haploid cells. Usually 3 of these haploid cells, they degenerate, and one which is left becomes the functional megaspore. The megaspore divides mitotically without cytokinesis, and 3 rounds of such divisions takes place without the cytokinesis, which result in the 8 nuclei structure. These nuclei are organized in a specific pattern, and finally cellularization of the embryo sac takes place, leading to the 7-cell structure or the mature embryo sac. It is the mature embryo sac stage which appear to be the most responsive for the gynogenesis. As I earlier mentioned that even if the ovules are harvested at the earlier stage of development, like maybe at the time when the meiosis is taking place, or at the time of when the mitotic divisions are taking place without the cytokinesis, the development of the female gametophyte continues till it acquires the maturity. And once it acquires the maturity, it is ready for the induction. Since it is difficult to identify the development stage of the gametophyte, therefore several external visible markers have been used or associated with the development stage of the gametophyte, like the association of the pollen developmental stage with the development stage of the female gametophyte. Then the size of the buds or the time with respect to the anthesis can also be used. So in this way, various markers have been or the various phenotypic markers have been developed that help to identify the developmental stage of the female gametophyte. However, these markers differ significantly among different plant species. The induction response also depends upon the health of the donor plants, or the developmental conditions around, or the quality of the female gametophyte at the time of induction treatment. Therefore, the donor plant should be healthy and should be maintained in a suitable growth conditions. Similarly, the seasonal effects have also been observed to impact the gynogenesis. The explants prepared from the florets that were developed first on the later branches usually are more responsive and show better induction as compared to the florets that appear later. Further, the explants collected from the medium-sized flowers of onion had a better induction response as compared to the explants harvested or explants that were collected either from the very large flowers or very small flowers. In some species, impact of the seasonal variations has also been reported to affect the gynogenic potential of the female gametophyte. So next is the pretreatment or the impact of the pretreatment on gynogenesis. Floral organs are usually pretreated to stimulate the gynogenic development, that is, the process of sporophytic development in the female gametophyte. As we discussed various treatment methods for the androgenic responses, from pollen like the temperature shocks, starvation, use of the chemical treatments, etc., these factors impact the gynogenic responses too. But for many of these factors, enough data is not available to make a consensus on the trends in responses. Therefore, we should consider these parameters too if we are setting up an experiment for the production of haploid plants through the gynogenesis. The application of low temperature shock is most successful for the induction of gynogenic responses in several plant species and is the most commonly used pretreatment. However, the high temperature incubation have also been used for induction, like in cucumber, when the explants were incubated at 32 degrees Celsius, it enhanced the production of embryos through gynogenesis. Further, the duration of the treatment application, like how long we need to give the treatment, the level of the pretreatment or the time of application, that is at what stage of the development it should be applied, these all vary considerably among different plant species. Like for sugar beet, incubation of the buds at 4 degrees centigrade is required for about 4 to 5 days to enhance the production of embryo through gynogenesis considerably. Whereas in rice, incubation of the panicle at 7 degrees centigrade for only 1 day is sufficient to induce the gynogenesis. Similarly, for the onion plants, when they were grown continuously or continuously maintained at 15 degrees Celsius, their female gametophytes were most responsive as compared to the onion plants when these were maintained or grown at either lower temperature of 10 degrees centigrade or at a higher temperature, that is about 18 degrees Celsius. So the overall pretreatment type, duration of the pretreatment, and the developmental stage of the female gametophyte when the pretreatment should be applied has a huge impact and should be considered carefully. Next is the impact of the media composition. The composition of the culture media is a critical factor, and their requirement varies at the different stages of the development. It is not possible to have only one media composition that works for all the plant species or all the development stages during the gynogenesis. Generally, MS salts and B5 media compositions have been used successfully for the gynogenic haploid production. Plant hormones, that is the auxins and cytokinins, are required at low concentration for the induction of gynogenic responses. Though in some species these responses have been reported without the use of the growth hormones exogenously, like in maize. However, the addition of the growth hormones at the low concentration enhance the gynogenic responses considerably, even in these species like in maize, where the gynogenic response was obtained without the exogenous supply of these hormones. So low concentration of plant hormones are helpful for the induction of these responses. Now the type of the growth hormone and its required concentration varies among the different plant species. Like 2,4-D has been successfully used for the onion, and indole-3 acetic acid or naphthalene acetic acid have been used in combination with benzyl aminopurine in beta vulgaris or helianthus species. Among various cytokinins, use of the benzyl aminopurine is usually most successful in most of the species. As a carbohydrate source, sucrose is most commonly used and is successful. Usually it is used at a 2 to 3% concentration. However, in many plants, much higher concentrations of the sucrose had produced better gynogenic responses. Like for the onion, up to 10% of the sucrose has been used successfully. And for maize, even the higher concentration, that is up to 12% of the sucrose in the culture media, produced best responses. Now, this higher concentration of the sucrose may not be required as a nutritional component, but may have some other roles that have not been yet identified. In case of the wheat, higher sucrose concentration has been reported to hamper the growth from the somatic tissues too, which is also helpful for the gynogenesis. Similarly, maltose has also been used successfully in plants as a carbohydrate source. Ovule culture is usually performed on the solid media, and agar is commonly used as a solidifying agent. So next is the origin of gynogenic plants. So generally the ovules are cultured at the mature embryo sac stage. And if it is difficult to extract or handle the ovules because of the size, because of their small size or maybe other regions, then either the ovaries or the complete flower buds can be used for initiating the gynogenic cultures. Irrespective to the type of explant, like whether the ovary, ovule or flower buds are used to initiate the in vitro cultures, gynogenic haploids originate from the female gametophyte. Female gametophyte in the angiosperm is mostly a 7-cell structure, and these are the cells which participate for the development of the haploid plants. Mostly the gynogenic haploids originate from the egg cell. However, these may develop from other haploid cells in the female gametophytes, like the production of gynogenic haploids from the synergid cells have also been observed. And it is the mature embryo sac that is more responsive. As earlier also we discussed that in many species, even if the ovules are harvested at the earlier stages of the female gametophyte development, its development continues in the in vitro conditions also till the maturity is achieved. That is, the mature embryo sac 7-cell structure is formed. And now the embryo sac is ready for the induction for gynogenesis. The overall gynogenesis process can be grouped into 2 stages, that is the induction and regeneration of the plants. So the first stage is the induction. Induction means acquiring the potential to form the sporophyte. That is the transition from the gametophytic developmental program towards the sporophytic development without the involvement of fertilization. Usually low temperature treatment is most commonly used for the induction of gynogenic responses. However, other factors like the chemical treatment, starvation, incubation in the light or in the dark can also impact the gynogenic responses. Pollination and germination of the pollen on the stigma also signal the female gametophyte to initiate the sporophytic development specific expression profiles. Therefore, early pollination or the delayed pollination can also induce the gynogenesis in some plants and has been used for this purpose. Further, the pollination with a defective pollen have also been successfully used for the induction of gynogenic responses. The idea behind the use of defective pollen is that though the pollen will germinate, it may grow on the female gametophyte and would release some growth substances that would diffuse from the pollen and prepare or induce the female gametophyte for further development. However, it would not be available for the fertilization with the egg cell or in the central cell. And for this purpose, pollen can be either exposed to the radiations or may be treated with the chemicals that will interfere with the normal development of the male gametes, and it would have a defective development. Next is the regeneration of the plants. So once the haploid egg cell or other cell of the female gametophyte like the synergids are induced for the gynogenesis, it initiates the cell division and further development starts. The regeneration of the haploid plants can occur either through the direct embryogenesis or through the indirect embryogenesis. So after gaining the induction, the egg cell divide and may directly start the globular embryonic development from here that result in the formation of proembryo, and this type of development where the cells divide and directly form the proembryo is called as the direct embryogenesis. Alternatively, multicellular tissue mass may undergo dedifferentiation and proliferate to form the callus. Later, the callus is induced for the morphogenetic differentiation using various combinations of growth hormones to regenerate plants. And this type of development that pass through the dedifferentiation and redifferentiation is called as a indirect embryogenesis. This dedifferentiation to form the callus may start at very early stages of the development, or in many cases the early divisions are very similar to the direct embryogenesis, but at the later stage it dedifferentiates to form the callus. However, the direct embryogenesis is desirable as the indirect embryogenesis may introduce unwanted mutations in the regenerated haploids. As discussed earlier too, the composition of the culture media, growth hormones, carbohydrate source, explant health, culture vessel, etc. can directly affect the plant regeneration, and therefore species specific optimizations needs to be performed. After the embryogenesis or organogenesis, the handling procedures for the shoots, excision of shoots, or the transfer of these excised shoots onto the rooting media, or the protocol to double the chromosome number of the haploids to produce the double haploids are similar to one what we studied earlier in the androgenic haploids. For more details on this procedure, refer to the first lecture. Though the gynogenesis has huge potential for genetics and crop improvement, there are several challenges for the in vitro induction of gynogenesis. The developmental stage of the female gametophyte at the time of culture may have significant impact on the induction of the gynogenesis. However, it is very difficult to assess the stage of the development of the embryo sac. Therefore, some external markers have been developed that correlate with the developmental stage of the female gametophyte, like the pollen developmental stage, time of the anthesis, or the size of the buds. However, these may differ significantly in the different plants and hence cannot be used universally for all the species, and species specific arrangements needs to be made. Dissection of the unfertilized ovaries for the female gametophyte is very difficult, and therefore ovules or ovaries are used as explants. However, the presence of lot of sporophytic tissue in these explants may complicate the developmental programs. Further, the presence of the few haploid cells in the female gametophyte and only a few gametophytes is another major limitation as compared to the anther or the pollen culture, where large number of pollen is available for the in vitro culture. Large proportions of gynogenic embryos degenerate at different stages of development. One possibility is that it happens because of the expression of lethal genes in the haploid stage, whereas in the normal development these are suppressed in the heterozygous form. So that's also a major challenge, that overall number of haploid plants that reach to maturity is very low. During the later development also, many deformities have been observed in the gynogenic plants, like the swollen hypocotyls, deformed cotyledons or the primary leaves, or the poor vigor are the common ones. And only few plants reach to maturity which are healthy and without any deformities. Further, the gametoclonal variations may appear that are undesirable, and extensive use of these haploids or double haploids in the breeding programs may reduce the genetic variations in the breeding germplasm. Finally, the application of gynogenic haploids. Haploids or double haploids are completely homozygous system and are very useful for breeding as well as for research purposes. It is very useful to dissect the genetic regulation of the traits. Usually the homozygous lines or the pure lines are generated by selfing for many generations, but through the haploid production it can be achieved in a single generation. Haploids are an ideal or convenient system to detect and study the recessive mutations. In haploids or double haploids, all the mutations are expressed and can be studied. Therefore, haploids can be useful to isolate the mutation that are usually masked in the diploid plants or the polyploid plants. During the meiosis that result in gamete formation, several new recombinations are formed, and haploids provides an opportunity to study the gametic variations which results due to the meiotic recombinations. Further, it is an excellent model to study the induction of embryogenesis and embryo development from the haploid gametophyte. For the many plant species which are self-incompatible, inbreeding is not possible, and therefore production of homozygous or pure lines for these self-incompatible plants is very difficult. Therefore, production of fertile homozygous double haploid plants can be very useful and can be easily obtained. Haploids are very useful for genetic mapping and genomic studies, and further the double haploids can be released as new cultivars too. For the genetic engineering based crop improvement programs also, haploids are very useful, as the chromosomal doubling of the transformed haploids can produce the true breeding transgenic diploid just in a single step. Though the androgenesis is more successful as compared to the gynogenesis for the production of haploids, but in some plants the gynogenic responses for the haploid productions are better and more successful as compared to the androgenesis. For example, the members of Chenopodiaceae or the Liliaceae family, where gynogenesis is more successful as compared to the androgenesis. The production of the gynogenic haploids is particularly important for the plants where either the androgenesis induction is unsuccessful or it is not applicable at all, like the male sterile plants which do not make any pollen grains or the male gametophytes, or the female plants in the dioecious plant species. So the androgenesis cannot be used for the haploid production, and gynogenesis is the only option in these plants. Gynogenic haploids carry the cytoplasmic traits of the maternal origin and therefore are useful for the maternally inherited traits too. Though the frequency of gynogenic plants that reach to maturity is very low, but these plants have higher genetic stability. Also, the rate of the albino production is very low in case of the gynogenic haploid as compared to the androgenesis. So overall, gynogenesis is a very important tool for the basic genetic studies and crop improvement programs. Though the androgenesis is more successful, but for the plants where either androgenesis is not applicable or if it is not successful, gynogenesis is the only option. So the gynogenesis has a huge potential, and in future accumulation of more research data, advancement and availability of the tools for female gametophyte harvesting, understanding of the mechanism of gynogenesis, and optimization of the protocol may help to improve or extend the gynogenesis to the recalcitrant plants. So in summary, in this lecture we talked about in vitro gynogenesis. We also talked about the various factors that are important for regenerating gynogenic plants, like the effect of the genotype, its plant health, pretreatment, or the composition of the media. Then we talked about various challenges that need to be addressed to improve the efficiency of the gynogenesis. And finally, we discussed about the various applications of the haploids and then specifically gynogenic haploids. Thank you.